All right, welcome back. Last time we looked at some of the key structures inside a cell, but honestly, we've only covered a fourth of it. There's a lot more going on in there. So, in this part, we're going to look at a few more organelles, their structure, and more importantly, what they actually do to keep the cell running. So, let's get into it. To make sense of everything we are about to cover in this video, let's recall this chart first. Organelles, remember, aren't just a random list of parts. They can be grouped by the job they're doing inside the cell, and that gives us four major categories. One group handles making and moving proteins around. We covered that in part one, so it's worth going back and watching if you've missed it. The link is in the description box. Today, we focused on the other three. There's a group that deals with storage and breaking down waste, then the energy organelles, and finally, the organelles for structure and support. So let's start with storage and breakdown organelles responsible for keeping the cell clean and toxin free. A lysosome is a vesicle containing hydrolytic enzymes that are used to digest macromolecules. They're not usually found in plant cells. The hydrolytic enzymes are produced by the rough ER and modified by the Golgi. You may remember this from the first video. And these vesicles that contain the hydrolytic enzymes can form new lysosomes, or they can simply fuse with an existing lysosome, releasing its contents into it. Lysosomes are called the cell's garbage disposal. Now, why are they called so? We will just discover that. They have a variety of functions. First, they fuse with food vacuoles to aid the breakdown of proteins, carbohydrates, lipids, nucleic acids into amino acids, sugars, and other monomers, which are then released into the cytoplasm to be used as nutrients, like you just see here. A lysosome fusing with a food vacuole, and then finally the contents are digested, waste products released outside by exocytosis, and the useful material is kept within the cell. So that's one function of lysosomes. Another function is that they act like a demolition team that takes down old and unsound buildings in a neighborhood. So what lysosomes do is they recycle cell components by breaking down worn out or damaged organelles. For example, if this mitochondria is damaged, lysosomes will recycle its components by breaking it down and releasing the useful materials into the cytoplasm of the cell. And this process is called autophagy, right? And the third function that lysosomes carry out is apoptosis. This is when a cell programs itself to die and to be destroyed. During apoptosis, remember, lysosomes release the hydrolytic enzymes into the cell's cytoplasm, and these enzymes start breaking down important structures like the cell membrane and other cellular organelles, and eventually the cell dies. This is programmed cell death. Lysosomes also play a role in destroying disease-causing organisms that might have entered the cell. The enzymes in lysosomes, remember, work best in acidic conditions. That's one requirement for these enzymes to work. So the next structure in this category are peroxisomes. Peroxisomes are small, round organelles responsible for breaking down fatty acids. They help neutralize free radicals and detoxify many toxins that may have entered the body. Alcohol, for example, is detoxified by peroxisomes in our liver cells, and liver cells therefore contain an exceptionally high number of peroxisomes. A byproduct of the reactions that take place in these peroxisomes is hydrogen peroxide, a toxic substance, and peroxisomes keep this toxin within them to prevent damage to the cellular components outside of that organelle. Therefore, peroxisomes are like mini sewage treatment plants. They help neutralize harmful toxins so that they don't cause chaos inside cells. Hydrogen peroxide is then safely broken down into water and oxygen. And then these byproducts are safely released into the cytoplasm. The last structure we have here is the vacuole. Vacuoles are relatively larger than vesicles, and they were also created by the Ruff, ER, and Golgi. They're fluid-filled sacs found inside cells and play an important role in storing water, nutrients, and waste products. So their main role is in storage. Vacuoles in plant cells are relatively large. The vacuole in plant cell also has a role in creating turgor pressure, which is simply the pressure exerted by the vacuole on the cytoplasm, and this pressure helps plant cells maintain its structure and keeps them turgid. So that was about the storage and breakdown organelles. Most cell activities depend on energy, whether it is building proteins, transporting materials, to simply keeping the cell alive. So where does all that energy actually come from? Let's look at the mitochondria and chloroplasts for this. The mitochondria are actually oval-shaped structures, and they're often called the powerhouses or the energy factories of a cell because they generate most of the chemical energy needed to power the cell's biochemical reactions. And this process is called cellular respiration. Cellular respiration involves the breakdown of glucose, right here, and in the presence of oxygen to produce adenosine triphosphate, the cell's primary energy carrying molecule. And oxygen is essential for efficient ATP production. Now, mitochondria are also distinguished by another feature. They have their own DNA and ribosomes apart from what exists inside the cell. Mitochondria, remember, are inherited maternally, and therefore mitochondrial DNA is also inherited only maternally. Mitochondria do not generate energy from nothing. They, in fact, convert the energy in the nutrient molecules, like the sugar molecule here, into a chemical form such as ATP that the cells can use. So, where in the cell is that energy originally captured to make nutrients like glucose in the first place? To answer that, we now look at another organelle, which is the chloroplast. Chloroplasts are oval-shaped structures found only in plant cells. And unlike mitochondria, which are present in nearly all eukaryotic cells, chloroplasts are only found in those cells that are capable of this process, photosynthesis. During photosynthesis, light energy is captured and it's converted to chemical energy in the nutrient molecules. This chemical energy is what cells use for cellular respiration. And in order to synthesize this glucose, you can clearly see that plants need carbon dioxide, they need water as raw materials, and then they produce nutrient molecules such as the sugar and an important byproduct, oxygen. Chloroplasts contain a green pigment called chlorophyll, and it is this pigment that is responsible for the absorption of light energy, and this is what drives the reaction of photosynthesis. Like mitochondria, chloroplasts also have their own DNA and they have their own ribosomes, distinct from what is found in the cell. In this sense, the mitochondria and the chloroplasts actually have complementary roles in a way that chloroplasts are capturing energy from sunlight, storing it in glucose, and while the mitochondria later break down this glucose to release energy in the form of ATP, because this is what the cells can actually use. Imagine if we were to remove all the organelles from a cell. Do you think it's the plasma membrane and the cytoplasm that will be the only components left? The answer to this question would be no, because the cytosol would still be containing organic molecules, ions, plus a network of protein fibers called the cytoskeleton. This network is what actually holds the cell together, supports the cell, helps keep the shape of the cell, and it also helps anchor organelles in place and assisting the movement of the cell and also movement of these materials within the cell. These fibers and filaments collectively are called cytoskeleton. So there are three main types of fibers: the microfilaments, the intermediate filaments, and the microtubules. So if we were to zoom in a little bit, so we see the microfilaments right here, the microtubules, and the intermediate filaments. Okay, so microtubules here are the thickest, these fibers right here. They are the thickest of the three. Microfilaments, on the other hand, are actin filaments and they are the thinnest. And intermediate filaments, as the name suggests, have a diameter that ranges between the two. So this is what the cytoskeleton is made of. It's a network of tubules and filaments. Now, the cytoskeleton isn't the only thing giving a cell its shape. Plant cells especially have another layer of support, one that animal cells don't have. And let's look at that next one, the cell wall. The cell wall is a rigid protective layer that sits around the plasma membrane or the cell membrane. So this right here is our plasma membrane and the cell wall is exterior to the cell membrane. Unlike the plasma membrane, which is flexible and partially permeable, the cell wall is firm and it is fully permeable, which simply means it allows water, ions, and small molecules to freely pass through. In plants, this cell wall is made of cellulose, a complex carbohydrate, and this is what gives this cell wall its strength and rigidity. And this is very important because when this vacuole fills up with water by osmosis, the vacuole exerts a pressure. Remember the turgor pressure? So as the vacuole exerts a pressure on the cytoplasm pushes against the cell wall, and the cell wall actually prevents the cell from bursting. And this turgor pressure is also what helps keep plant tissues firm and upright. So the cytoskeleton and the cell wall both give the cell its shape and support. But remember those microtubules we just talked about as part of the cytoskeleton? They're actually doing more than just providing structure and support. These microtubules are also part of structures like the centrosome, cilia, and flagella. The centrosome is a region near the cell's nucleus, and it is found in animal cells that functions as the cell's microtubule organizing center. As you see, the microtubules originate from the centrosome. It consists of a pair of centrioles that you see here. They are positioned at right angles to each other, and each centriole is further made of 9 triplets of microtubules, like you see here. The centrosome plays a key role in something almost every cell goes through during its lifetime: cell division. And just before the cell divides, the centrosome replicates itself. So we have 2 copies when the cell is actually dividing. So what these centrosomes do is that they produce fibers. They're called the spindle fibers. These fibers pull on the duplicated chromosomes to the opposite ends so that each cell has exactly the same number of chromosomes. So before a cell divides, remember the centrosome duplicates, then the resulting centrioles pull these duplicated chromosomes towards the opposite ends of the dividing cell, ensuring that each new cell receives a complete set of chromosomes. So that's about centrosome. So we've seen microtubules organize into centrioles to help pull chromosomes apart during cell division. Arranged differently, the same microtubules can actually push out of the cell surface. They could actually push out of the cell's plasma membrane, forming structures that let the cell move or move things around it. So now let's look at the cilia and the flagella. They enable movement. Flagella, singular, is flagellum. They are long protrusions And cells usually just have one of these, which are used to move an entire cell. So as the flagella moves in a whip-like motion, the cell is propelled forward. A classic example of this is a sperm cell. Cilia, on the other hand, are shorter and they are more numerous on the cell surface. They can actually move the whole cell or they could move substances along the outside of it. A good example of this is cilia that line the fallopian tubes or the oviducts, which help guide the egg towards the uterus, or say the cilia that line your respiratory tract. So they help to sweep mucus and trapped dust particles upwards towards your throat. So cilia, they move like a field of waving grass, all in rhythm, therefore pushing mucus towards the back of your throat. Together, cilia and flagella are mainly responsible for locomotion and moving fluids across the cell surface. But they're not just for movement. They can also act like sensory antenna, picking up signals from the cell's environment. All right, before we wrap up, let's put everything we've covered to the test. Here's the same cell diagram you've seen throughout this video, but this time the labels are gone. Pause the video here and see how many of these structures you can identify on your own. You could use this table on the right to jot down each organelle and its function. So go ahead, pause it now, give it an honest try, and then come back. Here's the complete label version. Check your answers against this. That's it for this part 2 series in cell organelles. If it helped, drop a like and subscribe for more breakdowns like this. And if you missed part 1, it's linked down in the description. Thanks for watching, and I'll see you in the next one.